Okay, Faye Hartman, a microbiologist at the School of Veterinary Medicine, is going to give a formal presentation, the first part of the time, to give you a little bit of information and history and background about MRSA, what it means. Okay, thanks very much for taking the time to show up this morning, and I will turn it over to Faye for the formal part of the presentation.
Well, thank you for coming. It's great to see so many people here to listen to us talk about methicillin-resistant Staphylococcus and the implications for us as researchers and what we're finding in the animal community. So just a little background, methicillin-resistant Staphylococcus aureus, or MRSA, are Staphylococcus aureus that are resistant to all beta-lactam antibiotics. That includes your penicillins, penicillin-beta-lactam combinations, and cephalosporins, except just recently there's a couple anti-MRSA cephalosporins that have come on the market for people. Methicillin resistance is mediated by a specialized gene called MRSA, which encodes an altered penicillin binding protein, hence why these organisms are resistant to beta-lactam antibiotics. The interesting thing about this gene is it's carried on a large mobile genetic element called the staphylococcal cassette chromosome. And in addition to carrying MRSA, this mobile genetic element is also an attractant for other resistance genes. So what we tend to find with these MRSA strains that we encounter is that they are extremely multi-drug resistant, and hence they're very difficult to treat compared to your regular Staph aureus infection. MRSA emerged in the early 1960s, and it happened to occur right after they introduced methicillin into the human healthcare community. And at that time, it became a leading cause of hospital-associated infections and now what we call healthcare-associated infections worldwide. In the early 1990s, another strain of MRSA emerged called CA MRSA, or community-associated MRSA. Does anybody remember hearing about in the media the football players that came down with MRSA infections or the wrestlers that came down with MRSA infections? That was the emergence of this community-associated strain where we're seeing MRSA infections in people that had no contact with a hospital or an old age home or any other kind of healthcare setting. Currently, MRSA has taken another change in its epidemiology, and we're seeing community-associated MRSA in healthy young adults and children, which is very unusual. In addition, these strains are starting to come into the healthcare community, and we're seeing these community-associated strains causing healthcare-associated infections. One of the core contributors to MRSA infections is colonization. Now, this is true of Staphylococcus aureus, too. Approximately 30% of us are colonized with Staphylococcus aureus, and so when we get an infection, it's usually due to Staphylococcus aureus for skin infections. If we look at the general population in terms of MRSA, between 1% to 5% of the general population in the US is colonized normally with MRSA. However, this can change depending upon the different groups of people that you're looking at. A recent study found 38% of HIV-infected individuals colonized with MRSA. If we look at healthy Texas college students, 7.4%. And then if we look at an Ohio homeless shelter, 25%. So depending upon the community setting that a person is living in will predict the prevalence of MRSA colonization in that group. So I've thrown out a couple terms here, HA MRSA, CA MRSA. That's one thing that you'll notice in the literature and the news when people are talking about MRSA. Well, the jargon has become more complicated. We have hospital onset, community associated, community onset MRSA, and we have a new one called LA MRSA. Has anybody heard of LA MRSA before? Anyone? LA MRSA stands for livestock associated MRSA. So it looks like now we are starting to see the emergence of MRSA in animals. In fact, we have had reports of colonization infections in a wide variety of domestic animal species, including cats and dogs, cows, horses, pigs, birds, rabbits, turkeys, the list goes on. And what we see in terms of transmission is most of the strains that we see in dogs and cats are coming from people and are being transferred to animals. This is also true for horses and cows and pigs. But in addition to that, we are seeing host-adapted strains of MRSA, such as in pigs and horses, that normally like horses and pigs, coming in and now colonizing and infecting people. So if we start by looking at the food animals, we're going to look at pigs first because the pig-MRSA relationship has a very interesting story. It started in the Netherlands, and if we look at the Netherlands has a very low prevalence of MRSA in its human population, less than 1%. In fact, they have an intensive search and destroy policy there where they screen everybody coming into a hospital. If you have MRSA, you're isolated. And what they were finding back in around 2003 and before is that the people who came into the hospital that were colonized with MRSA were people who traveled, foreign travelers. Well, what they found in 2004 was a girl and her family were colonized with MRSA, no history of travel, and when they looked at them, they found out they all lived on a pig farm. So in 2005, Voss et al. Started doing a survey of people who worked on pig farms and lived on pig farms and found a 760 times greater prevalence of MRSA colonization in those people compared to the general population, which is huge. And it turned out that this strain was sequence type or clonal complex type 398, or what we call livestock-associated MRSA. They then did a study looking at pigs, and in Dutch pigs in 2005, they found it in the pigs, which makes sense because they found it in people in close contact with pigs. And they found a colonization rate of greater than 50%. Really, it's up to 80 to 90% of pigs in the Netherlands are colonized with this LA MRSA. What type of disease does it cause in pigs? Well, interestingly enough, it causes a very minimal amount of disease. It'll cause rare skin infections, rare causes of mastitis and urogenital infections. And when they look at colonization, it's throughout the entire production phase. So as soon as piglets are born, they become colonized from the sow. And all through that production chain, they can find colonization in pigs. The interesting thing about this strain that they're finding in pigs is that it appears to be a host-adapted strain, an older strain of Staph aureus that used to infect people that got rid of all of its human virulence factors and ended up settling in the pigs. If we look at people, the people that have frequent contact with pigs definitely is a risk factor for carriage of MRSA. A German study, they found 86% of people working with pigs were colonized with this LA MRSA. If they looked at their family members, 4.3% of the family members. If they looked at the veterinarians, 45% of the veterinarians handling the pigs and 9% of their family members. And as you move out from contact with pigs, you lose the colonization relationship with LA MRSA. If we look at disease in people, we will see infections. It's a very minimal amount of infections, most often skin infections. However, there have been a few reports of severe invasive infections, and usually these strains of LA MRSA have acquired back some of the human virulence factors. And over in Europe, there have been a few hospital outbreaks due to LA MRSA. So the importance of this is people who work with pigs are at a higher risk for becoming in contact with MRSA. They can be colonized. And so if you have to be hospitalized or you have an infection, it's important to let people know, I have pig contact, I may have a MRSA infection. So do we have anybody in here doing research with any pigs on campus at all? A couple people, so we'll revisit those a little bit later in the presentation. What about other countries? Is it isolated just to the Netherlands? Well, it isn't. They found it over in Asia, other European countries, and also in Canada. And recently, it's been identified in the United States as well. 45% in Iowa swine and swine workers. Ohio, they only found a 1% prevalence. And then in North Carolina, a 45% prevalence. I think it's always been there. It's just we never started looking for it until after 2004 and 5 when Voss conducted that study in the Netherlands. And so more studies are being done in the US just to see, what is the prevalence in our pig population here in the United States. What about other food animals since we're seeing it in pigs? Well, the Europeans, again, 8% of Dutch farms, which are veal calves, found that they were positive for colonization with MRSA. You can find it in poultry, you can find it in dairy cows, and you can find it in cattle. The interesting thing about dairy cows, though, it's a very uncommon cause of mastitis, which is interesting considering Staph aureus is a common cause of mastitis in cows. We see mastitis due to MRSA very infrequently. However, there's a new strain of MRSA coming out over in Europe. It contains MEC-C instead of MEC-A, and that strain seems to be causing an increasing number of cases of mastitis. So maybe eventually we may see that over here as well. When you have farm animals that are colonized with MRSA, you get a spillover into any other animals, just like into people on that farm, coming in contact with that organism and potentially becoming colonized. I have on here teaching animals, just because I'm from the veterinary school and we use teaching animals quite frequently. And a colleague of mine in Colorado looked last year at a group of pigs that they got in for one of their surgery classes, and 80% of the pigs that they brought in for teaching purposes were colonized with MRSA, which gives you a whole new perspective on what type of infection control procedures should you have in place so people working with those pigs don't become colonized. So that gives you a little bit of an idea of what we're seeing in some of the food animals. What about companion animals? As I mentioned, we're seeing it in all domestic animal species. And so if we look at horses, horses is another interesting group of animals because the initial reports in horses were very sporadic, and we only saw it in hospitalized horses. In fact, here on campus, we were one of the first people to report finding MRSA in horses. However, those reports have been on the rise. We're seeing it in the community. We're seeing it in equine clinics. And it causes a variety of infections, usually wound and surgical site infections, which is similar to what we see in people. And the prevalence can vary. New York found 4.7% of farms. And when they looked at clinics, 16% of horses coming into the clinic were colonized with MRSA. What does this mean for people? The strain that seems to be infecting horses, at least here in the US, is a USA 500 strain, which is a strain of MRSA that really no longer infects people quite as much as it used to, but it seems to enjoy residing in horses. It readily moves between people and horses. And in studies, they found that 9% of hospital personnel, veterinary hospital personnel working with horses, can be colonized. And so horses, too, can be a community reservoir of MRSA. If we look at MRSA that we see here on campus in terms of our horses, we have a very low incidence of MRSA infections in the equine population that we see here at the veterinary school. And in fact, in 2011, I started a study to screen every single horse that came into our practice. And after 6 months, we quit the study because we didn't find one horse colonized with MRSA. So for our equine population here, at least in central Wisconsin, we seem to have a very low prevalence of MRSA in our horses, which is very good. So what about other companion animals? Let's move on to the smaller species. How many people in here work with dogs or cats in their research? How many people have dogs or cats as pets? All right. 62% of us have a pet, and there's over 78 million dogs and 86 million cats. So that's a lot of dogs and cats in the United States. And we do see MRSA in dogs and cats. And we also see MRSA in exotic species in birds and rabbits. In fact, rabbits seem to be very prone to Staph aureus infections and MRSA infections. And so if we look at dogs and cats, mainly what we see are the hospital or healthcare associated and the community associated strains of MRSA that we find in people causing infections in dogs and cats. So it's a reverse zoonosis. They're getting it from us. The disease that we see, again, is usually complicated wound infections, post-surgical site infections, and sometimes we'll see a skin or an ear infection. What about colonization? Is it the same as what we see in people? Since, you know, Staph aureus loves people, do we see colonization in dogs and cats? And really, we do not see a permanent colonization in dogs and cats. It's more like a transient organism popping in, causing some problems, or not doing anything, and then leaving. And in fact, Loeffler conducted a study over in Europe where he looked at Humane Society. Dog came in with a MRSA wound infection. He sampled all the dogs in contact with that dog, and several of those dogs also had MRSA present in their oral cavity and the nose. Once that wound infection was cleared and the dog was off antibiotics, they went back in and screened that dog and all the dogs housed with it, and they couldn't find any MRSA. So it's a very transient organism in dogs, at least, and cats. However, there is a concern, especially for me over at the veterinary school. There is a risk of hospital-associated transmission. So if we have a case of MRSA in the hospital, we need to make sure that we take appropriate control measures so it doesn't get spread to any other patients in the hospital. If we look at the number of MRSA isolations we've seen in dogs and cats here on campus at the teaching hospital, it's been very small. Here we had a little spike between 5 to 6 isolations in that year, and when I did pulse field gel electrophoresis to see if the strains were related or not, none of those strains were related. They were all independent isolations. And so it'd be interesting to see, because I imagine that what we had at that time point was a spike in MRSA colonization in people, too, in the area, and their pets. And then this year again, we had 4 cases of MRSA infections out of dogs. So still a very small number when you see about a different methicillin-resistant staph species that we'll talk about in a little bit. So what about research animals? This is why, of course, everybody is here. What are we seeing in research animals? We know that MRSA can go from us to animals and from animals back to us. Well, interestingly enough, there is hardly any information in the literature about MRSA infections in the research community. I was able to find 3 papers in the literature, both of them in rhesus macaques. This here non-Staph aureus infection is like a coagulase negative, like a Staphylococcus epidermidis, methicillin-resistant staphylococci that caused a skin infection. And so I sent out an email to CompMed just to see, if anybody else in the US had been having any issues with MRSA, and I got a couple replies back. Mostly everybody in the research community doesn't have an idea. Most of the replies were from primate facilities, and 3 of the 4 primate facilities that got back to me did notice that they were having issues with MRSA infections in their primates, non-human primates, with implants. Another group had a non-human primate that had a wound infection, and one group got back to me that had a dog that had a MRSA infection, but they blamed it on the teaching hospital that they sent the dog to get a surgery done. So, which was kind of easy to do, I imagine. What about the campus prevalence? What are we seeing currently on our campus animals? So this is a representation of all the isolations that we've had to date. We didn't have any prior to 2009. Then in 2009, we had 2 cats with head implants that developed MRSA infections in those implants. In 2012, this represents 4 non-human primates. One of the non-human primates had a skin infection, and they sampled the other 3 primates that are group housed with it. And those other 3 primates were colonized nasal cavities and oral cavity were positive for MRSA. In 2013, they sampled another 4 primates different than those previous group of primates because one primate had a severe wound infection, not an implant. And they sampled the monkeys that were housed with that primate, non-human primate, and they also were colonized with MRSA. And then we have this one case that Drew was referring to of a dog in 2013 that was positive in its blood. It had a catheter placed for MRSA. And then down here, just recently, we had 4 pigs that were sampled on campus just to screen and see, well, since MRSA is so prevalent in pigs, do any of our pigs on campus, are they colonized with MRSA? And we found one of the 4 pigs was positive for MRSA. However, we do not know what strain it is. The typing for that strain is still pending. It could be a hospital-associated or a community-associated strain of MRSA. We do not know for sure yet if it's one of the actual livestock-associated MRSAs, and we'll find out hopefully within the next 2 weeks. So as I mentioned, there are some other methicillin-resistant staph species that we need to be concerned about, and that's primarily the methicillin-resistant staph intermedius group. Consists of pseudointermedius, intermedius, and delphinii. The one we mainly see is the pseudointermedius, and it is a main commensal organism of dogs and cats. Intermedius can be found on horses. However, we are finding methicillin-resistant strains, and it's the same mechanism as MRSA with the MEC-A gene and the staphylococcal cassette chromosome. There's another organism that we see primarily in humans. This is also, I'm finding it in dogs and cats. It's a Schleifleri subspecies coagulans that can be methicillin resistant. And it is an emerging problem in dogs, cats, horses, and people, these other strains of methicillin-resistant staphylococci that we're seeing. If we look at the isolations, if you remember, think back to the MRSA isolations that you saw on campus for the different species. In 2013, I had over 100 isolations of methicillin-resistant staph pseudointermedius in dogs. 3/4 of the dogs with pyoderma coming in, are due to MRSA infection. Ear infections, wound infections, post-orthopedic surgical site infections. So it's a huge concern. And in fact, I was very surprised when we had that dog isolation that it turned out to be a MRSA and not one of these methicillin-resistant staph pseudointermediate strains. It does cause severe infections, just like MRSA. These are two very severe wound infections that these dogs contracted, and they both survived. And for people, very few reports of infections, but people in contact with dogs and cats that are colonized with the methicillin-resistant staph pseudointermediates can also be transient colonized. What about healthy dogs? The interesting thing about healthy dogs, I don't see it in healthy dogs. It's only dogs on therapy, dogs that are sick, dogs that are immunocompromised. And the reason I mentioned this strain too is those last group of 4 primates that we sampled, one of the primates was colonized in the oral cavity with MRSA, but then it had a methicillin-resistant staph intermediates group in the nasal cavity. So we do have to be aware there's other methicillin-resistant strains of Staphylococci out there that can also potentially colonize or cause infections in our research animals. And so one of the most important things that we need to consider about these organisms is because of that drug resistance, if an animal is colonized, they're going to be more likely to develop an infection. Doesn't necessarily mean that they will, but we want to try to break that transmission cycle. And the transmission of MRSA is a contact transmission. So it's either contact with someone or animal that has an active infection, contact with a colonized individual, or contact with a contaminated object or environment. Because Staphylococci tend to like to reside in the oral cavity, the nasal cavity, or the skin, we're shedding epithelial cells all over the place. It's very easy for the environment to become contaminated with these organisms, and they can survive for extended periods of time in the environment because they're Staphylococci. They have very thick cell walls, they resist desiccation. And it's been shown that our hands are the most common cause of transmission of MRSA between humans, animals, and each other. So as I mentioned, colonization is an important consideration because it puts you at an increased risk for infection. Doesn't mean you're going to get it, but you are at an increased risk. So when you're colonized, you want to know, as a person, because when you go in, if you have to have a hip replacement, of course, they're going to treat you differently, do some decolonization procedures because they've shown that people are more prone to develop an infection. It's the same with animals. If an animal is colonized with MRSA, we need to be careful because we don't want to put them at risk for developing an infection. And also, we need to be careful with our antibiotic therapy. It's been shown that use of fluoroquinolones and third-generation cephalosporins has been associated with an increased risk for acquiring a MRSA infection. Extensive wounds and invasive procedures, which makes sense because you're compromising the natural infection barriers of an animal or a person. As we mentioned, contact with positive animals, humans, or the environment, especially in a research setting or a hospital setting. We need to control that so we don't transmit it to other animals that we may be working with. Hospitalization, definitely. That's why we hear so much about hospital-acquired infections. It's a different kind of setting, close contact, hard to control hygiene practices and transfer, so another setting for an increased risk for transmission. Immunosuppression, pretty obvious, but one of the biggest risk factors for developing a MRSA infection, especially if an animal or person is colonized, is foreign material, suture, implants, catheterization. Very big risk factor if you are colonized or come in contact with MRSA or Staph aureus for that matter. So as I mentioned, hands are the leading cause for transmission of MRSA. So one of the biggest things that we can do is enact properly performed hand hygiene. And so that's one of the measures out of this group that guideline was developed was to introduce proper hand hygiene on campus to everybody working with research animals. In addition, as I mentioned, rational use of antibiotics, that's in terms of being careful with the third-generation cephalosporin usage and fluoroquinolone usage. Aseptic technique, we all know how important aseptic technique is. Sterilization and disinfection procedures. Here's a big one. Everybody working with implemented or surgical procedures or animals that have wounds or are at higher risk really need to understand, contact transmission guidelines, hand hygiene guidelines. It's very important. And of course, our environment, keeping our environment clean and disinfected. And one of the biggest things, because of the way Staph aureus or MRSA is spread, is that we look at our frequent high-touch surfaces. Has anybody seen any of the articles coming out about doctors and their stethoscopes being contaminated with MRSA? Cell phones, money. So what we do at the VMTH and are hopefully going to help everybody else here in their laboratories to do is to institute using disinfectant wipes. The disinfectant wipes are great for decontaminating high-touch surfaces like keyboards, doorknobs, phones, anything that is frequently touched to keep that microbial load down. Another thing that's important is if you do identify an animal that is positive for MRSA, to make sure that you institute appropriate contact precautions with signage for everybody so they know, okay, this is the PPE I need to wear, this is what I need to do to transport this animal, this is how I clean and disinfect this environment. We use these at the teaching hospital for any animal that comes up positive, so everybody going into that room or handling that animal is aware of what precautions they need to take so that we don't transmit that organism to another animal or contract it ourselves working with that animal. So in summary, we're seeing MRSA in a large variety of animal species. MRSA is flowing from us to animals, and the animals are sharing it back with us. For some animals, it's very interesting because they've acquired a host-adapted strain. So this LA MRSA prefers, the larger food animals, and the horse strain prefers horses, yet those strains can come back and also colonize and cause infections in us. And so out of all of these discussions, one of the things that we're hoping will be helpful to everybody is we've developed some hygiene plans, a fact sheet about MRSA, so if anybody has questions about MRSA, as well as appropriate hand hygienes and other documents. And I believe this sheet is going to be passed out to everybody. Yes? In the back of the room. So at this point, if anybody has any questions, we'd be happy to answer them.
I have a couple of follow-up items of information to give to everybody. And the first is that, as mentioned in the presentation, that healthy animals, healthy dogs and cats, don't seem to get a MRSA or antibiotic-resistant infection and keep it for a long time. But there's a couple of keys here that are really pertinent to the type of work that we do. Is that at-risk populations include immunocompromised animals and any type of animal with a chronic implant, a catheter or any type of implant. And immunosuppressed animals and animals with implants are common in the research world. So we have a lot of those on campus, and those are high-risk populations for MRSA. So that's a reason why we have to be very careful about this and be aware of that. And it's easy for a human being to give MRSA to an instrumented or immunosuppressed animal and then have that animal give it back to somebody else. So we have to be aware of those high-risk populations that we deal with. And as an area of emphasis, the single most important thing that anybody can do to prevent transmission is proper hand hygiene. When you're working with animals or anywhere around animals in these rooms, wear gloves, wear gloves whenever you can, and do proper hand sanitization. Just keep your hands clean, and that is the best thing that you can do. Okay.
I will mention too, those two animals, the two cats. The other important thing to keep in mind is if an animal does get MRSA infection, it could be detrimental to your project. The one cat did not survive. The other cat did survive, and the dog mid-project also did not survive the bacteremia. So you spend all this time and this money on these projects, And if an animal develops one of these severe infections, it might be at the end of that animal for that project. So that's another important thing to keep in mind for trying to control and prevent transmission.
Yeah, and I would clarify that these animals didn't just get sick and die, from this. The decision was made to humanely euthanize them. So they didn't, they didn't survive, but they didn't just die from the infection. Have a question?
Is there any epidemiology on MRSA in rodents?
I'm glad you asked that question. That's very important to us. There is none that we're aware of. We've searched, there is none, but that is something we want to find out. So we are in the early stages right now of initiating a campus-wide study to look for the prevalence of MRSA in rodents. We're in the early stages of that. We hope to do that. We hope to get that data, which will be very useful, not only for us on campus here, but throughout the research community worldwide. So we are going to embark on that. We're going to get some data and let everybody know. So what we want to do, and I ask for everybody's help on this, is what we want to do primarily is identify the most likely target populations on rodents on campus that could potentially have this. This would be any rodent with any type of chronic implant, catheterization, implant, anything of that nature. And what we'd like to do is basically just take a swab, just take a swab around the implant site, and then we just test it and grow it up and see if it's there. So we're looking for rodents with any type of chronic catheter or instrumentation, especially if you're working with an immunosuppressed or immunocompromised strain of rodent, and many of those are around. So whether it is a rodent or any other type of species, what we want to do is identify chronically implanted or instrumented animals, immunocompromised animals. If you have any in your colony, you'd like to be part of this, let us know afterwards. Just come up and see us afterwards, and we'll work to make arrangements, because this is data that's important for everybody. I'd also say, it was brought up in the presentation, is that pigs are a source of concern. And pigs that aren't instrumented, or aren't necessarily immunocompromised, seem to be good reservoirs of these antibiotic-resistant organisms. So if you work with or have pig colonies, that's another target population you want to look at. So we will be doing this shortly. We could use your help in identifying appropriate target populations. We'll get that data as soon as we can. We'll get it published. We'll get it out there. That's important information for people to know.
Yes?
So what about the people that are colonized? What are the consequences of knowing that you're a carrier
So,
for your work or for trying to prevent
right.
it to your research animals?
Right, and so that's a very good question. There's different camps out there, even in human medicine. Do you identify all your carriers or don't you? Because of the, whether or not that's appropriate in confidentiality. And so even if you're a colonized individual, as long as you're taking appropriate hand hygiene and other hygienic practices, it's not an issue. It should not be an issue. And if you're a colonized individual, just because you're colonized does not necessarily mean you're going to develop an infection. It's just like, more of us are colonized with Staph aureus. And most causes of pyoderma in people is Staphylococcus aureus, because that's part of our normal flora. Most infections that people and animal gets, it's, your normal flora gone crazy in a spot that it's not supposed to be. And so just because you're colonized, it doesn't necessarily mean you're going to develop an infection. However, if you're going into the hospital for some reason, some major surgery, they probably, especially if it's an orthopedic surgery, they will screen you, because it has been shown for orthopedic surgeries that if you're colonized with MRSA, you're more likely to develop an infection, and so they'll do decolonization procedures. But that's kind of the only place right now in the US where they're actually doing some active screening of people. Yes?
I have like a two-part question. So my understanding is that people are colonizing their nasal passages. First, I guess I just wanted confirmation that that is the case.
So the question is, are people just colonized in their nasal passages? And No, they are not just colonized in their nasal passages. So just doing nasal swabs, you can miss a small percentage of people. The big places for people to be colonized are your nasal passages, your oral cavity, groin, and armpits, and perianal area.
So my second question is how important is it to use like surgical masks or something like that when dealing with catheterized animals or something like that to try to prevent infection that way? But you're telling me that's not the only method, so we're not sure if there's any information on reducing spread.
Yeah, and so the only information out there is looking at human catheter procedures, and most of those procedures that they're looking at for human catheterization procedures, it's using aseptic technique gloves and prepping the area appropriately. It doesn't say anything about, they don't require people to wear a mask for doing those. Yes?
Are you concerned about implants with exteriorized wires and tubes only, or are telemetry implants also included?
Let True answer that question.
I'd say mostly those with exteriorized components to it. We'd still be interested in, let's say, osmotic pumps, that are put in animals and it's all sewed up, so there's nothing exteriorized. But we'd still be interested in that, especially if they are an immunocompromised strain, in any way. Just to see if any part of the process of the procedure, the surgical procedure itself, allowed some of the organisms to get in. So we would have interest in that.
Part of the reason for doing this prevalence study, since there's so little information out there regarding the prevalence of MRSA in our research animals, is once we know how much prevalence there is, maybe there isn't a lot of prevalence, which means great, these are the measures that we need to institute. However, if we find a higher prevalence than what we expect, then we'll have to institute different measures so that we can make sure that we can protect the animals and the people that are working with them.
Who will be the primary contact for the rodent prevalence study?
Me, you can. Again, if you don't know me, I'm Andrew Jeffcoat, the senior program vet at the School of Medicine. You can look me up on the directory and send me an email or see me afterwards.
Are the sentinel animals routinely tested for MRSA, the rodents?
No, we haven't done, we have no idea. We haven't done any. I mean, even in the non-human primates, the only ones that we've cultured are ones that have been group housed with another animal that developed an infection. And we haven't looked at any of our implemented non-human primates, and based on those 3 other facilities in the US that definitely experience problems with their non-human primates with implants. It's very interesting. Yes?
And Faye, the only known route of transmission, human to human or human to animals or animals to human, is direct contact. There's no airborne transmission through breathing in MRSA that has been identified thus far, right? It's only
direct contact.
Direct contact only.
Right.